Assalamu alaikum guys, hope all you are fine. In this video we are going to discuss how the HCL is created in your stomach and how it helps in the digestion of the food. So what is the function of HCL? That is released in the stomach. The HCL created acidic environment in the stomach and helps the pepsin to work and help in the protein digestion. It means HCL is main function is to help in the digestion of the proteins, and HCL main function is to kill, to inactivate, to paralyze the bacteria that enters with the food, because the food we intake has some bacteria in it. So it can cause infection in our stomach, in our duodenum, in our GI tract. So we have to kill these bacteria. So HCl acid is that chemical which kill the bacteria from our food. So three main types of cells in our GIT you can see here is neck cells, parietal cells, and there are peptic cells. So we one by one discuss the function of their cells. Number one, peptic cells. Peptic, as the name represents, peptic cells secrete pepsinogen, and you know pepsinogen is for the digestion of proteins. Pepsinogen break proteins into amino acids so they can be easily digested and used by our body. Peptic cells are also known as the chief cells. Second cells, which are neck cells. Neck cells secrete mucus for the lubrication of the content to reduce the friction. Parietal cells are also called as oxantic cells, and their main purpose is to secrete our gastric acid, which is hydrochloric acid, which I have already mentioned you the functions of hydrochloric acid. And second thing which is released by parietal cells is intrinsic factor. So intrinsic factors help in the absorption of the vitamin B12. So if we discuss the parietal cells, because in this video our major purpose is to describe, to explain the structure of parietal cells and the secretion of HCl. So these parietal cells you can see here secrete an acid solution which contains 160 millimeter of hydrochloric acid. So the pH of this acid is 0.8. It is highly acidic, so it helps in the digestion. The parietal cells you can see here has the caniculi. What is the purpose of these caniculi? You can see here the caniculi have the folds which increase the surface area for the absorption, for the production of this acid. So these parietal cells having these caniculi helps in the production of the HCl, which is acidic to 0.8 pH. Now, how this HCl is formed? We have discussed that parietal cells form HCl, but how? The main pump that is responsible for the production of HCl is hydrogen potassium pump. First of all, the potassium which is moving out of the cell, and the hydrogen which is moving out of the cell. From where this hydrogen ion comes? This hydrogen ion is formed by the splitting of water in the cell. The water split up into the hydrogen and hydroxide ion, and this hydrogen comes from this water breakdown. Now what happens is that the carbon dioxide which is coming from the bloodstream combines with this OH and forms a carbonic acid, and you know carbonic acid is very unstable. The chloride ions which is moving out of the cells, the carbonic acid follow it and move out of the cell, and the chloride ion moves in passively. This chloride ion is transported into the lumen where it combines with the hydrogen, and this hydrogen and HCl combine to form HCl, which is the hydrochloric acid. So I hope you will understand this, how HCl is formed. It is the hydrogen ion which comes by the splitting of water, and the chloride ion which is moving into the cell and then into the lumen, so they form HCl. In consent with the chloride ion, some sodium also move in, and this sodium combines with the carbonate, this bicarbonate, to form sodium bicarbonate. Now, which factors stimulate the acid secretion? So you can see here I have written the three things: the acetylcholine, the gastrin, and the histamine. You know, acetylcholine is a neurotransmitter and it is released by an autonomic nervous system. An autonomic system, in which autonomic nervous system? The parasympathetic stimulation. You know, parasympathetic stimulation always increase the activity of GIT. So this acetylcholine helps in the secretion of pepsinogen by the peptic cells, hydrochloric acid by the parietal or the oxantic cells, and the mucus by the mucus cells. So the both now this side, gastrin and the histamine secretion, they increase the HCL secretion by the parietal cells, but they have very little effect. Major role is by the acetylcholine. So they help in the positive effect. They give the positive effect to GIT and increase its secretions. Factors which inhibit ACL production or HCL secretion are enterogastrone, GIP, VIP, and secretin and glucagon from the duodenum. These two play major role. Secretin and glucagon from the duodenum inhibits the HCL production in the stomach from the oxantic or the parietal cells. The summary. So you can see here there are three phases of the HCL production. When you see a food, just smell, your vagus nerve activate the parasympathetic stimulation, which excite pepsin and acid production. Number 2 phase, gastric acid. When you eat that food, your local sensory reflexes, your gastrin histamine production increase HCL secretion. And your third one, which is the intestinal phase. So your nervous mechanisms, your hormonal mechanisms, when the food enters the intestine, it increase the gastric secretion, gastric acid secretion like HCL. So summary of this video. In this video we have firstly discussed the cells type, which includes 3: parietal, oxantic, and the mucus cells. Then we have discussed the mechanism how an HCL is formed, and number 3, the factors affecting the secretion, positive regulation and negative regulation. So if you have got that point, if you have not get any point, you can ask me in the comment. And if you like this video, then must subscribe to the channel.